Assalamualaikum and very good morning to everyone. Today, me and my group are going to present about rootless hair as a reliable source of forensic genetic information. The main reason why rootless hair is traditionally being considered unreliable is because conventional forensic testing depends heavily on PCR amplification of STR markers. According to the journal, the DNA found inside rootless hair shaft is extremely fragmented and usually too short for successful STR amplification. As a result, many hair samples from crime scenes were either ignored or only used for mitochondrial DNA testing, which has lower individual discrimination power. This created a major forensic limitation, especially in cold cases where hair evidence may be the only biological material available. What makes this research highly unique is that the researchers successfully extracted usable nuclear DNA from single rootless hair shafts. Instead of relying on STR profiling, they used advanced sequencing methods optimized for degraded DNA and focused on SNP analysis. The study demonstrated that even highly fragmented DNA from rootless hair can generate enough genome coverage for accurate genotype calling and forensic identification. This is extremely important because it opens new opportunities for solving cold cases and identifying unknown individuals using evidence that was previously considered unusable. This brings us to our primary case problem. Shed hairs are found at almost every violent crime scene, yet forensic analysts have historically discarded rootless hair shaft for nuclear DNA testing, relying instead on low discrimination mitochondrial sequencing. Why? Because as hair shaft elongate, an endonuclease called DNase 1L2 aggressively degrades the nuclear DNA into tiny strands. As a result, almost all DNA fragments found within a hair shaft are shorter than 100 nucleotides. Because standard PCR STR assays require intact DNA strands spanning several hundred base pairs to create amplicons, traditional forensic typing on rootless hair fails completely.
In this study, the researcher analyzed 50 head hair samples and 30 pubic hair samples to determine whether rootless hair could still provide reliable forensic DNA evidence. The study found that most DNA fragments were shorter than 100 nucleotides, explaining why traditional STR profiling frequently fails in rootless hair samples. Despite severe fragmentation, the recovered DNA was mostly nuclear DNA, averaging about 96.94%, while only around 3.06% was mitochondrial DNA. This finding is highly significant because nuclear DNA offers much stronger individual identification power compared to mitochondrial DNA. Therefore, the major forensic challenge is not the absence of DNA, but whether current forensics technologies are capable of recovering and interpreting highly degraded genetic material effectively. To critically evaluate the forensic value in rootless hair, three realistic identification approaches must be compared. Firstly is traditional STR profiling, which remains the forensic gold standard due to its speed, extensive validation, and compatibility with criminal databases. However, STR analysis depends on longer intact DNA fragments for PCR amplification. Since the study showed that most hair DNA fragments are under 100 nucleotides, STR profiling becomes unreliable for rootless hair evidence. The second approach is mitochondrial DNA analysis. This method performs better with degraded samples because mitochondrial DNA exists in much higher copy numbers. However, mitochondrial DNA only traces maternal lineage, meaning multiple maternal relatives may share identical profiles. As a result, its discrimination power in criminal identification is limited. The third approach is single-stranded next-generation sequencing with SNP profiling, which is the method proposed in this study. Unlike STR analysis, this method is specifically optimized for fragmented DNA recovery and successfully generated accurate SNP profiles, even from low-coverage hair samples. The study also demonstrated that this method can support both direct identity testing and investigative genetic genealogy.
Based on the findings of this study, the recommendation is that forensic lab integrate SNP-based sequencing into their existing forensic workflow when dealing with rootless hair sample. But we are not suggesting that SNP sequencing should completely replace the traditional STR analysis. STR profiling still remains as the global forensic standard because it is faster, cheaper, and it already accepted by the court system. Instead, SNP sequencing should be used as a secondary method when STR analysis fail due to degraded or fragmented DNA. In this journal, it demonstrates that rootless hair sample produce genotype concordance above 99.4%, which indicate that SNP profiling can generate reliable results. Next, to implement this method, forensic labs should follow a structured workflow. First, rootless hair evidence must be collected from the crime scene and preserved to prevent contamination. Next, specialized DNA extraction technique must be used because the DNA fragment within the hair shaft is highly fragmented. The extracted DNA then undergoes library preparation and sequencing that allows scientists to recover genetic info that cannot be analysed using the STR analysis. Finally, the SNP profile generated from sequencing can be compared with reference sample for identification. The implementation of SNP sequencing for rootless hair evidence could strengthen forensic investigation. Why? Because first, it increases the amount of usable biological evidence. Instead of evidence being discarded, rootless hair can now provide genetic info that can help move an investigation forward. Second, it improves opportunity for solving cold cases where hair evidence may be the only remaining biological sample. Next, it provides stronger investigative leads through genetic genealogy approaches, which can help forensic scientists to narrow down the potential suspect when other methods are unsuccessful. Finally, it expands the overall capability of forensic lab by transforming previously overlooked evidence, like this rootless hair, into a valuable source of genetic information.
To conclude, the final outcomes of this landmark research are fundamentally redefining the limits of forensic molecular biology. Cup and his co-authors have shattered the historical misconception that rootless hair shafts are genetically useless. By successfully generating up to 3.86-fold coverage from head hair and 4.47-fold coverage from pubic hair, they have proven that a simple, automatable workflow can unlock highly accurate nuclear SNP profiles from highly degraded cell-free hair fragments. The real-world significance of this methodology cannot be overstated. In practice, the authors note that they have already actively deployed this exact technique to generate successful investigative leads for violence criminal investigations, and the identification of unidentified human remains across unresolved cold cases. However, with this massive technological leap comes a critical responsibility. The researchers explicitly warn that using this method outside of tightly controlled law enforcement context would breach the terms of service of genetic genealogy platform. Therefore, our group emphasized that future implementation must be accompanied by robust policy frameworks and stronger legislative disincentive to prevent unauthorized genetic surveillance and safeguard civil privacy. Ultimately, this paper shifts the balance of power back toward justice. It ensures the millions of shed hairs sitting silently in old evidence lockers can finally speak. As the authors eloquently state, it is now entirely possible to uncover investigative leads and establish absolute identity using nothing more than a few centimeters of a single rootless hair. That's all from us. Thank you for watching.
I just had to let you know you're